and I'm going to be introducing Dr. Joseph Kirschvink, and he received his PhD from Princeton University and is the Nico and Marilyn Van Wingen Professor of Geobiology at Caltech. He originated the snowball Earth concept to explain weird features on Earth's oldest glaciations, discovered the tiny magnets that animals use for navigation, and has recognized several major shifts in Earth's spin axis that have driven biological evolution. And today his talk will be, could the terrestrial biosphere have started on the flanks of Tharsis? Welcome, Dr. Kirschvink.
Oh, great. And I'll start by asking Bob my question from the previous talk. The original Mars helicopter, Bob, where'd you go? Oh, yeah. The original Mars helicopter idea, you were going to use a Samsung cell phone architecture of some sort. Now, Qualcomm, whatever. Most drones on Earth use GPS navigation coupled with a magnetometer. Now, you don't have GPS in Mars. Are you going to keep the magnetometer on board?
It's probably there, but I don't know what's on the chipset.
Please, keep it.
We have very high powered magnets powering our rovers at just close proximity. So I don't know where we get.
Well, actually, we've checked some of those motors, and the flux on the rare earth magnets are so closely coupled, just a few centimeters away, you get perfect signals. Mind you, we tried to get a magnetometer on Mars 2020 and on Curiosity and failed on both rounds, even though we had $3 million matching from private sources. Anyway, that's an aside, but as you may gather, I am a magnetist. I study magnetism, I live and breathe it. I'm also a biologist, and as mentioned in the kind introduction, I am fascinated by small magnetic objects and things. One of my lab projects at the moment is unraveling the biophysics of the magnetic sense organs in animals, which actually I think may have evolved on Mars initially. So we'll make that drink. So what I would like to focus on in the brief 25 minutes I have today is whether the terrestrial biosphere could have started on the flanks of Tharsis. And this is kind of a nutty idea, but not so nutty when you think about it. The real title, an analysis of the electrochemical environment of early Martian high-altitude glaciers during late Noachian time. And the reason this is important is that we know Mars has been throwing rocks at us for 4.5 billion years. We have on the order of 200 Martian meteorites. Some of them, using magnetic studies as well as argon studies, we know got here on low temperature trajectories. They're not heated. So phrasing it in a little different way, Mars has been spitting at Earth for 4 billion years. It has implications for planetary protection as well. On the other hand, if you realize that, and you say, well, if Mars ever did evolve life, if life somehow had either gotten to Mars or evolved there, the natural transport mechanisms are there. Those Martian meteorites get to Earth without heating. And so the problem of the origin of life cannot be divorced from Mars. If it had evolved on Mars, it would be here, it would be us, and we might not know it. We probably wouldn't know it. So that actually makes an interesting prediction in terms of the environment of Mars. But when you step back from that a little bit more, and you say, well, compare Earth and Mars, which I'm going to be doing here very quickly, why might Mars have been a better place? And there's a very simple, and I think a very powerful argument when you look at the early environment of Earth and Mars that argues Mars might have been a better place. And it comes down to electrochemistry. So very quickly, Earth and Mars, life is here now, no visible life on Mars. There's a deep gravity well on Earth. It makes it very difficult for meteorites to get ejected from Earth. We have tektites, they're molten materials, but we don't have any ammonites with fusion crusts showing they were ejected and they came back. We have 200-odd Martian meteorites. It's easy to get off of Mars with an impact. We have a horrible rock record actually on Earth of the time where we think life evolved. It's been buried, it's been burned. On Mars, it's there, it's excellent. That time interval where we think life evolved somewhere is preserved on Mars. So here's really what I'm going to tell you. Electrochemically, I mean, I'm wearing a biological hat as well as a geological hat. Was the original metabolism on Mars, not of Mars, but of life, sloppy or precise? And if you look at the scenarios for the origin of life, particularly where people think Mars may have, or life may have evolved in a weird, restricted, extreme environment, I raise my hand and say, if you look at what lives in those extreme environments on Earth, they didn't evolve there, they moved there. The way natural selection works, Darwinian evolution, is you take a sloppy system and you make it better. If you apply that reasoning to the fundamental metabolism that drives all life and all cells, you want an ancestor that starts in a very sloppy, energy-rich environment. So my focus on life, don't look at Europa, don't look at Enceladus. The electrochemistry there is minimal. Look for a place where we have wide chemical variability. And I found a place. It's on Mars. It's the Tharsis province. And in fact, looking at that environment 4 billion years ago, 4.5 billion years ago, solves a hell of a lot of the problems we have trying to figure out how life evolved. So I'm going to focus on Tharsis. I'm going to worry about ozone in the atmosphere, very elegant studies by geochemical groups in San Diego, and then where it might be. So here we go. Bear with me. This is a science talk. So life is driven by oxidation and reduction reactions, and if you can see my little pointer moving around here, if you have a half reaction here that dumps an electron this way, you gain energy. If you go the other way, you lose energy. So all metabolism on Earth drives this way. So when you look at those coupled reactions and look at the biochemistry, you find that there are electron transport processes that sit and dump electrons from highly reducing to highly oxidizing. And each little step generates enough energy for a molecule of ATP or so, or NADPH or something else. And it kind of works like this. You go bump, 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 boom. The chemistry is fairly complex and highly evolved. We start with highly reduced, and then boom, you get down eventually to oxygen. That's this redox tower thing I showed. So here are the two kind of ideas. If you go back to the classic biochemical ideas, people think, well, this probably evolved stepwise. Norm Horowitz at Caltech thought this many years ago. And the idea is that you would have a little bit of energy from some redox jump. You can grab a little molecule of ATP, and then as time moves on, you add a little thing to the end of that chain, and it goes on, and you end up with this highly complex biochemical pathway. Problem here, that little system has to be efficient at the start. If you're going to get, sulfate to sulfite electrochemical reaction, you need very, very precise, highly evolved enzymes to do that. That's not going to be the way life could start. Evolution takes a sloppy system and makes it better. So what I would prefer is to find an environment somewhere in the solar system where you have large gradients, maybe going from oxygen to methane, huge chemical availability, and you can have a sloppy system. Boom. What evolution then will do is take gene systems, duplicate it, tailor it, tinker with it, and you can start extracting more and more energy. So you go from that to this to something like that, and then end up with that system. So, of course, the first time I suggested this, people said, oh, oxidizing conditions destroy prebiotic molecules. Why do you want the origin of life in a place where there's oxygen? Stanley Miller couldn't do it. Yeah, Harold Urey could not do that with lots of oxygen. But there's a way of doing this. As a geologist, you know that oxygen is sequestered, you get redox stratification. And you can get basins or puddles that interact with oxygen. So I'm going to go to Mars really quick. By the way, this is why I don't like the concept of life in extreme environments, as I said. They moved there. If you do the genetics of something at hydrothermal vents or in highly high thermal environments, those bugs moved there. They didn't evolve there, okay? Sorry, NASA, you're spending too much money on life in extreme. Anyway, that's not where it's from. Can we find an early environment with large redox gradients? And I think there is one. Let's go to Mars. Here's a view of Olympus Mons. If we go back in time, well, just today, we know that there's a dichotomy in Mars. The northern basins are very low. The southern highlands are very high and old. We know why it's there. Tharsis is the largest gravity anomaly in the solar system. Basically, when it erupted, the mantle of Mars had stopped convecting. It was solid. So the Tharsis province stays there with a huge gravity anomaly. And as you spin around, it has to go on and stay onto the equator. And this is really unusual. The mantle of Mars stopped convecting before the volcanic pile accumulated. It holds up the weight, and it hasn't collapsed in 4.5 billion years. Tells you something. Tharsis volcanoes did not come from the mantle of Mars. They came from a heat source near the core mantle boundary. That's an important point. It means the volcanics equilibrated with nickel iron metal. The average elevation of Tharsis is also 28 kilometers above the base. That's another critical thing, because that's how you get the oxidants on Mars. Okay, so that's the background on Mars. Two very critical things. Geology and geochemistry, we've known since Mariner that there are valley networks, streams, things. If you go back about 20 years to a beautiful paper by Phillips, Murray, and Zuber, Solomon et al. In Science, they actually looked at the MOLA data, the Mars Laser Altimeter data, and recognized that Mars, because it is solidified, Tharsis came out, it rolled to the equator, it warped Tharsis. But that warping, these valley networks drain downhill from the warp of Tharsis. It allows you to date the valley networks. And what it tells you, by the way, excuse my Japanese, I'm half-time in Japan and half-time in Tokyo, and I can't, they have two sets of slides running around, but anyway. So anyway, during Noachian time, prior than about 3 billion years ago, these valley networks are old. You can date them because of this warpage. But the amount, and that really constrains Tharsis to come out in a very short period of time. And if you worry about the volatiles that are released, you get something like 120 meters filling over the planet, or up to 1.5 kilometers of volatiles, water on the North Polar basins. So the other thing, those gases that came out in that period of time were equilibrated with the deep Martian mantle. No plate tectonics on Mars after that point. There may have been a little bit earlier. That makes some very important geochemical arguments. First off, I won't go into it too much. We know that there is water. My colleague at Caltech, Bethany Allman, has done beautiful work showing that there's a wet phase that's been originally completely reducing, then oxidizing. We know, as Mike mentioned earlier, that there are beautiful evidence for flowing water, rounded stones from the Curiosity rover, et cetera, et cetera. I won't go into that. But the point I want to make is that the Tharsis environment is precisely what Harold Urey and Stanley Miller worked on about in the early 1950s. It's a pity, as I'll get to, they did a lot of experiments with reducing gases and spark mixtures, and they could get all sorts of organic carbon compounds there, nucleotides, amino acids, fatty acids. But then somebody told them, but those are not the gases that come out of volcanoes, they're more oxidizing. When they tried those gases, it didn't work, and it kind of threw things for a knot. But if you go back and look at those geological oxygen buffers, I'm going to use a few terms here. This is now temperature versus log oxygen for gas, basically the equilibrium amount of oxygen. And when you go to geology and they say oxidizing, it doesn't mean oxygen, it means how many minus log units the oxygen is there, which is on this thing. So we have the iron woostite buffer from native iron to iron oxide, phalite, magnetite quartz, and then magnetite hematite there. I'm going to show you something interesting that came out of the Deep Carbon Observatory studies a few years ago. This beautiful paper by Wetzel et al. Turns out if you look at this oxygen fugacity, now this previous slide is here, oxygen fugacity. These buffers, they're shown on the horizontal line here, relative to the iron wüstite. Earth's mantle is very oxidized. It turns out when you have a planet the size of Earth, the pressure deep in the core takes the reducing materials and disproportionates it and makes it oxygen reducing. And so the Earth's mantle is oxidized. The gases that come out of Earth's mantle dominated by CO2. If you go to more reducing equilibrium environments, you start getting things like methane, ammonia, hydrogen, primary gases coming off. And so the Martian mantle, actually measured from meteorites, comes this way. Lunar mantle is even more reducing and so forth. So the point I'm trying to make is that the classic Miller-Urey reduced gas mixes would have come out from the Tharsis volcano. It had to, the way that plumbing is sourced. So Earth is up there. The moon-forming event also sucked out a lot of the native iron and sucked it to the Earth's core. That didn't happen on Mars. So if we go then to the prebiotic synthesis argument, certainly the Urey-Miller experiments were there. The gases coming out of Tharsis would have been dominated by methane into NH3 and so forth, the reducing materials, very much so. They're in the summit of these volcanoes, very cold temperatures with huge lightnings. I mean, any volcanic eruption is accompanied by copious lightning, simply from triboelectric separation. So you've got a Urey-Miller synthesis place. On top of that, remember, my whole goal is to find places where we have redox chemistry. So what about the oxygen? I refer you to a beautiful paper by Jim Farquhar, Mark Tiemens, and Theresa Jackson back in 1998, about 20 years ago. By looking at the oxygen isotopes in the carbonates from this famous Martian meteorite, ALH 84001, you can actually get a fingerprint of the oxygen going through ozone. There's a mass-independent fractionation of the oxygen isotopes, where oxygen 17 and 18 are fractionated differently with respect to oxygen 16. And that's a fingerprint of going through ozone with an ultraviolet stage. A quarter of the oxygen atoms that are in those carbonates were up in the Martian atmosphere as ozone and circulating through. So there was an ozone layer on ancient Mars. Absolutely beautiful, stunning, because here we've got the volcano with reducing gases coming out, and yet they go into an atmosphere with ozone. So the question, can you mix those two? And it turns out if you look at Earth today, we can actually know where the ozone layer is and scale it. We can actually go back and change the scale height for ozone on Earth to match that on Mars. So here's Tharsis on Mars. Average elevation of the plateau, 28 kilometers above the base of the North Polar Basin. So the question is, where would the ozone layer have been on ancient Mars? And you can do a first-order calculation on this. You compare the atmospheric scale heights, H is the scale height, and then you worry about the 1/E exponential decay of the heights. And you scale that with the background Boltzmann constant temperature and so forth. The critical numbers from Earth and Mars are like this. Molecular mass, atomic mass units for the atmosphere of Mars is higher, it's got more CO2. You want to then change this to Noachian time, going back 4 billion years. You keep that CO2 needed for the greenhouse, at least the atomic mass units. You want to raise that temperature up to something where water could form, say 260. And then you do that scaling, and what you estimate is about 13 kilometers for the scale height of the atmosphere. We go back then to our Tharsis, put that scale height on, and what you discover is you get about 25, 26 kilometers. So you go back to Noachian Mars, put the atmosphere back in, you walk up into the ozone layer on Tharsis. It hits the ground. So you have these volcanic gases coming out from the eruptions hitting ozone. So what does that mean for the origin of life? How can I bring this in? We know that Tharsis had glaciers. There are actually Amazonian glaciations that are preserved on Mars. There's good evidence for Noachian glaciers on the flanks of Tharsis. So here you've got a high volcano, erupting volcanic gases. You have the lightning hitting the Urey-Miller gases coming out of the volcano. What happens? It freezes onto the surface. When the eruption stops, the ozone percolates in. My gosh, you've got a redox gradient that's unlike anything else we've ever seen really in the solar system. Maximum. So, and if you think about the stratigraphy of those glaciers, you can worry about top to bottom. The bottom, and you would get a stratigraphy. You say, you go on that, look at what you would have prebiotic organic mush with volcanic ash, ice-rich water vapor just condensing, and you would build up a stratigraphy where the ice is trapped, maximum redox gradients. This flows down hills and will make. So this is the only place in the solar system where I've seen a maximum redox gradient present, predicted to be there. We don't know how long it'll be there, but it's there. And so, bang, you go from highly reducing to highly oxidizing right in that thing. So, well, those glaciers are going to flow, they're going to go down, and what happens? What about actually producing the origin of life into environments? Now, every Precambrian geomicrobiochemist has their own favorite idea for the origin of life. But one of the things we know about the biosphere today is that it is based on nucleic acids, particularly ribose DNA and deoxyribose, ribose in RNA and deoxyribose in DNA. One of the worst problems in the origin of life has been getting ribose. Ribose is part of the backbone of DNA. You need to be able to get it somehow. And there are kind of a couple of mechanisms that have been proposed. It's been difficult. Making these little sugars abiotically is not easy because they like to form tar, coal tar, basically. And it's just they like to polymerize. And there are two solutions that have been proposed to this. Steve Benner's solution is, gee, if you have an environment with lots of calcium and borate, you can actually use it to generate these carbons. Sutherland had another solution by making two other molecules photochemically and splicing them together. And these groups have been warring back and forth. Benner, for example, is concerned that Sutherland's solution doesn't work without graduate students somewhere making chemicals and bringing it back. And we don't think there were graduate students on Mars or in the ADN. Anyway, that's a battle that geochemists can go back. But the Benner model is nice because it does make a prediction about environments that are consistent with Mars. And basically the idea is that you need the calcium and the borates to stop the further reactions when you get to this stage in the reaction synthesis. And the calcium borate minerals like colemanite and ulexite and a few other things are common in environmental things. And by the way, you also need a little bit of oxidized molybdenum to do it, which would be present in things. But there's important things about these borate minerals. They mainly form in dry lake beds and deserts, and they dissolve easily in water. So you want an environment where you can get them down. Death Valley is a good one. We have chains of craters that produce it. Mars is another good environment. Chains of craters where those go through. We actually landed the Opportunity rover on environments. Unfortunately, you didn't have a way to measure boron, which is a pity. But that's an environment. I mean, so this is what Steve Benner accuses me of. Move it to Mars. Okay. And seriously, so how much time do I have? 2 minutes, but he started 10 minutes late. Okay, fine. Anyway, so we know that life can move. Impacts on Mars will produce meteorites, splatter them in all directions. ALH 8411, we know came from Mars. It has the Martian atmosphere in it and the isotope anomalies. We do have very interesting techniques to prove this. We did this about 20 years ago. We have a scanning magnetic microscope, which actually can go and resolve magnetic anomalies at a few micron levels. We used this with Ben Weiss and others, my former student is now at MIT, to show that this famous meteorite actually got to Earth without being heated even to 40 Celsius. And I'll skip over to this one. Turns out, if you look at just the natural remnants, you can see magnetic anomalies. When you heat it, they go away, even by 40 degrees Celsius. The rock has not been magnetized. And it went from Mars to Earth without being heated even to 40 Celsius. So what I will do is then skip a little bit to say, since that there is one of the original arguments about this meteorite that still is viable. This is McKay et al.'s famous paper, launched the Astrobiology Institute and all sorts of things. Worm-like fossils were one of the ideas, 4 ideas, but the one that still is there is the magnetite in those carbonate blebs on this meteorite. Many of them, 10%, look like they are biological. And we've actually constrained, in the study that's just been done, that turns out we can test the origin of the magnetites, whether they're detrital or chemical. Detrital if it's biological, chemical if they're detrital. And the answer is actually complex. I'll summarize this by saying that we've shown that the magnetites themselves are detrital, but they had to form in a very strong magnetic field, almost like 10 Gauss, 20 times stronger than the Earth's field. So it's very difficult to think about how to do that on Mars, but that is the solution to that. And so I will just come back and, anyway. I'll conclude by saying I think we might be Tharsians, not just Martians, and maybe Freeman Dyson is the example Tharsian. So to keep on time, even though I'm 10 minutes short, I'll take questions. Okay. Just the guy in the green shirt.
It's a little off your talk, but you're an expert in the effects of magnetism on biology. So some of the ideas for protecting against galactic cosmic radiation involve extremely strong many Tesla magnetic fields around astronauts. What do you think about a strong magnetic field affecting the health and nervous systems of human biology?
I'm actually not free to discuss that at the moment.
Well, that's a bummer.
We have manuscripts in review that, unfortunately, the editors do not want us to speak about that. No, but the thing is, you do want to put astronauts on Mars in a locality where you could use some of the local magnetic anomalies to deflect cosmic radiation. Unfortunately, all of our attempts to get magnetometers on the rovers have failed, damn it. Although InSight will finally have a magnetometer out down on the surface. And really, if you can put, for 1 milligram, a magnetometer on your iPhone, most of the questions that you can answer could be done. The data that we had from the ALH 84001 is now arguing that the magnetic field when those carbonate blebs were formed may have been 10 Gauss. You could do that on Mars conceivably. The distance from the core to the surface of Mars is about half of that on Earth. The field drops of 1 over r cubed to get a factor of 8 to 10 difference. Not inconceivable, even though my colleague David Stevenson cringes when I ask him about that. I think a proper magnetic survey would help locate future habitable sites. You want to do it in a place where the intrinsic magnetic anomalies on the Martian surface would deflect those. And I will say that I think you do not want humans to spend 2 years in space without a background magnetic field comparable to what you have on Earth. I'll say that much.
Doctor, this is strictly a speculative, personal opinion question. How far along did life advance on Mars? We know we found perhaps little simple single-cell organisms. How far along do you speculate that life developed on Mars?
My friend Norm Sleep once suggested that eukaryotes evolved on Mars. All right. My gut feeling is that if you look at the history of life on Earth, it has been limited by the oxygen concentrations. Early Earth, we know, was less than 1 part per billion oxygen. We have detrital grains of pyrite and uraninite that survive the entire rock cycle, from erosion in the mountains to the distal toe of a marine fan. And the 10-micron sized grains would be gone if there was a part per billion of oxygen in that environment. You can't get higher life without oxygen. The metabolic energy is not there. On Mars, we have the reducing materials. We had oxygen produced photochemically, ozone in the atmosphere. I was joking when we started putting landers down, look for clamshells. We don't know how fast life can evolve or not. But I mean, certainly I would not be surprised to see stromatolites, algal mat structures. But we really, Earth seems to have choked and slowed down the evolution of life. And a planet which has the natural geochemical gradients there might have been much, much faster.
Okay, another question?
I was noticing the structure of hemoglobin and chlorophyll are remarkably similar, only one has magnesium and one has iron. And both of those are derived, from magnesium iron type of silicates. Could you comment on that, suggesting the ubiquitousness of life?
Well, the heme biochemical synthetic pathway is fundamental to both hemoglobin and the chlorophyll molecules. In fact, there's an anaerobic version of the heme synthesis, which is the same in anaerobic and aerobic organisms and feeds into both heme and chlorophyll. There are 7 minor substitutions that have been seen in the aerobic version of that synthetic pathway. So no, there's a clear biochemical similarity between those. What was the point of your question, though?
The question suggests that since magnesium silicates are a very common thing out in the universe, I mean,
Oh, well, yeah.
see lanes of dust everywhere in the galaxy, it suggests that magnesium iron complexes could serve as the basis for forming these 2 very important molecules, and suggests, of course, the ubiquitousness of Earth-type life.
Would love to. I would love to see hemoglobin on Mars.
You will.
Okay.